Okay, and so now I'm going to be talking about cytotypr and how to read in the data and do some initial gating. So I have a lot of stuff written here that you can kind of go through in a little bit more detail as you're trying to gate and analyze your first flow cytometry data. But all of the examples really comes from this paper that we published in Scientific Reports, which is Cytofeature Engineering, a pipeline for flow cytometry analysis to uncover immune populations and associations with disease. And it goes through a lot of the information about flow cytometry, how everything works, and shows a lot of the plots that we're going to be seeing today. So if you want more information, this paper has some really good descriptions of the data and what you can do with it. And so the goal of cytotypr is to identify flow cytometry immune cell populations. And for a description of how to actually stain the cells, we have another paper, Current Protocols for Acquisition of High Quality Spectral Flow Cytometry Data. And so this protocol describes in depth how to prepare your single cell suspension, how to count your cells on a flow cytometer, and do all the different staining and the proper controls. So now to actually analyze the data, flow cytometers have the standard file format called FCS files. And that includes information about cell measurements and metadata describing, when the samples were taken, on what type of flow cytometer, and it gives the medium fluorescent intensities, or MFIs, of the fluorescently conjugated antibodies or fluorescent probes. And so typically a different FCS file is created for each sample. So when we're analyzing the data, we want to first start by installing all of the different packages that are necessary. So we'll install all of the packages that we need onto your local computer. And this installation process right here only needs to be performed once. Now when we go, I'm going to be showing a full example of how to obtain some basic results and plots for your flow cytometry data using FMOs. And so it's going to start by reading in the flow cytometry files, performing initial gating, and then performing the feature engineering and plotting the results. So since we've already installed the packages here, we now need to load them in so that they're accessible for us to use. So we just use this library function and we call each of these individual packages here that we're going to be using further. Now when we read in our data into R, we're going to have multiple FCS files that are generated from an experiment, and we're going to read these in as an NCDF flow set object. We're first going to start by identifying the folder where all of our FMO files are created. And the way that we can do this is I have this R markdown file saved into this protocol folder. And so to navigate to where all of our FMOs are saved, this ... means go up one folder. So I'm going to go up a folder and we can see all of my folders here. Now to navigate to my FMOs, I just write out the path to this folder. So if I go into my inst folder here, then to my ext data folder, and then my FMOs, now it has a list of all the FMOs. And we can see we do full equals true, it'll write the path to each of these individual FMO files that we can call here with FMO FCS files. So then we're going to read them into an NCDF flow set here. So we're taking the location of all of these FMO FCS files and we're going to read them into a flow set. And this may take a minute or so to do. Just kind of a note, the best practice for acquiring samples means naming marker channels the exact same name every single time in an experiment. But sometimes if you have experiments that are months apart, you may forget the exact wording of the channel that you used the very first time. So in this case, at one of the time points, I named one of the channels Zombie NIR with a space and all capitalized NIR. At a different time, I named it Zombie and then lowercase NIR. And so if those aren't standardized before we read them in, it won't recognize them as the same marker channel because of the different capital case here. Or in this case, another option was if you have an underscore NIR. And so we need to standardize this. And the way we can do this is with this channel alias function. And so we write in, we create a data frame where alias is zombie space NIR-A. And so this is what we're going to convert all of our zombie NIR channels to. And this is saying, if we see zombie underscore NIR, if we see zombie capitalized NIR, or zombie space lowercase NIR, it's all going to be changed to the standardized format right here. So now that we've read it into this NCFS FMO, we can see we have an NCDF flow set with 20 different samples. where each of the row names are the different files that we have. So this is CD103 FCS file, CD122, and the column names are all of the different fluorescent marker channels that we see. So we have, Alexa Fluor 532 and so on. And we can also index this NCFS FMO. So this NCDF flow set right here has 20 different samples. If we want to look at the first sample, for example, we can use these brackets and then 1. And so here we have all the information. So this is just one sample for our CD103 FCS file. So we can look at just one file at a time if we're interested. So after reading in our data, we're going to want to do some of the initial gating strategies. So this is really similar to what I was talking about in the last video, where we'll first gate on singlets or single cells, lymphocytes, and then live cells. And so we use the OpenCyto package, which uses reproducible algorithms to gate cells based off of marker density. So we can use a variety of different gates that OpenCyto has already created. So we can look at this OpenCyto. This is just their website. I've added a link in this example as well. And it gives all the different options for gating methods. So when we're gating our cells, we generally start with a singlet gate. So there's a singlet gate that already exists that we can call and it will gate on our singlets. And then we often use min density. So it takes a minimum density between two different peaks here and calculates the center. So in this case, it would be here. In this case, it would be here to gate on cells. And so we often use this for our lymphocyte gating and for our live gating. So what we're going to do is create a CSV file that has the information for all of this initial gating strategy. And so here we can see this is our gating. And so we have our alias column here. And so this is what we're going to call each of our different gating steps. So here we have 3 different gating steps. We have our singlets, our lymphocytes, and our live cells. From there, the next column is population. So when we do a positive population, it's going to take the information to either to the right of the gate or inside the gate. So in the case of a singlet gate, when we go here, if we do positive, it's going to take all the cells inside the gate. If we had done a negative, it would take only the cells outside. When we're using the min density function here, if we do positive, it's going to take everything to the right side of the gate. If we do negative for population, it's going to take everything to the left side of the gate. The parent is which population we're gating from. So we're always going to start with root for our very first gate because that's going to take all of the different cells. After we've gated on our singlets, then the parent is going to be the singlet population where we're going to gate on our lymphocytes. After that, we're going to gate the remaining lymphocytes and gate on our live cells. The dims is the dimensions that we're going to be using for each of these different gating arguments. So in the case of our singlets, we can use forward scatter area and forward scatter height, and we're going to be using our singlet gate as I described earlier. I often use a wider gate equals true as a gating argument because the normal gating is really, really small and removes a lot more cells. And I set this max iterations at 20 because if you do more, it will just run iteratively and take a super long time. For the lymphocyte gate, we're going to look at forward scatter on our x-axis. I'm using the min density function. And if it's not gating in the correct place, you can add this gating range. So generally for our cells, we see the lymphocyte gate placement somewhere between 4 times 10 to the 5th and 8 times 10 to the 5th. So you can kind of guide it a little bit here. And then lastly, for our live cells, we're gating on our zombie NIR dimension using the minimum density function. And for our zombie NIR, the gating range is generally somewhere between 1 times 10 to the 4th and 1 times 10 to the 5th. So we have our gating strategy here. If you want some more information about how to create a CSV gating template, this OpenCyto website, I have a link again, has more information here about how to gate and use these different CSV gating templates. So here again, I'm going to navigate to the correct folder for the gating strategy CSV file. And then this is a way that with this fread function, we can view the gating strategy and just check that everything is read incorrectly. So again, this is the same thing that was shown with the CSV file that I have pulled open. Then we can read in the gating strategy to a gating template object with this gating template function. And then if we want, we can plot it. So here we can see that we're taking our root population. We're using a singlet gate to gate on our singlets. Then we're using a minimum density function to gate on our lymphocytes and then our live cells. From there, we are going to want to actually apply the gating set to our FMO samples. And so we have to convert our NCDF flow set, so this is our NCFS FMO, to a gating set object. So we use this gating set function. And from there, we can apply the initial gating to the data. And so this is going to see, we can have the output here, which shows gating for singlets, lymphocytes, and live cells, and then it's finished. And again, this may take a minute because it's gating all of your different samples. And then if you want to check how the gating looks for a specific sample, we can use autoplot, index one of our samples here. And so we can see our singlet gate with the percentage of cells that were gated, our lymphocytes, and then this is our live cells. So then we want to convert our data into a flow set object and convert that into a data frame so that we can use the tidyverse set of tools in R to further analyze the data. So here we're going to pull out the live node from the gating set. So this is the last node in the initial gating strategy. And we're going to use the gs_pop_get_data function here to pull this out. And the output is a cytoset object. So then we convert the cytoset object to a flow set object. Flow set FMO gated data is a flow set now. And now we're going to convert it into a data frame using the tidy flow set from this package cytotypr. And so here, what's shown below is just a little bit of a example of what the FMO gated data looks like. So we have our file name column, which has, in this case, CD103 FCS file, and it has all of the different columns for the different fluorescent markers that we have in our sample. So in this case, each row represents a different cell with the various marker channels as the columns and the corresponding MFI values for each cell in the marker channel. So now that we know how to read in our data and gate it, be sure to check out the next video on how to actually feature engineer the data.